The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, best digital health, best incubator accelerator, best startup.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Welcome to the Galien Foundation Week of Innovation webinars here in New York. The objective of this week is to provide additional context and background of the largest annual event, the Prix Galien USA Awards, to be held tomorrow here in New York. In conjunction with the annual Galien Forum of speakers presenting scientific experts and the leaders in global health, we have developed a new webinar format that as an example that we are doing today, to give nominees for the best pharmaceutical, best biotech, best medtech, and best digital solutions, the best startup in the incubator category, to provide some first-hand visibility to the product nomination through these webinars that we have here. Now, this year, we received a record 147 nominees in 6 categories from 126 companies covering almost 15 therapeutic areas, ranging from cancer, from neuroscience to infectious immunology to rare genetic disorders. So we'll be closely watching this today in this webinar. Now, this webinar is about the pharmaceutical category, which is the focus of today. The webinar is 1 hour long and that we have been allocated. And we have 5 panelists here and nominees for the solid tumors that I'll quickly introduce in a second. And the purpose of this webinar is really one central fact that we're trying to uncover. With the success of these novel gene therapies that we've seen advancing the survival timelines in blood and bone marrow cancers, can that be replicated in solid tumors? And here with me, I have 5 nominees that will describe their innovations to say that truly can be translated back to solid tumors, what we've learned from the hematology-based cancers. So let's move on to our discussion. Let me first start introducing with our first panelist that I can see here is Greg Freiberg, who's a Vice President in Medical Affairs in the LMAC region from Amgen. Greg, can you please describe your product and the innovation that you're driving?
Yeah, thanks, Sanjay. And first, let me thank the panel for inviting today. It's an honor to be on this panel with this all-star crew. My name is Greg Freiberg. I'm a medical oncologist by training, though I've been working in industry now for about 17 years. I've had the good fortune over the last 4 years of being involved in the development of Lumakras. Really, this is not just a story of the molecule. I think it's a story also of the pathway, of the KRAS pathway. And anyone, of course, who touches solid tumors understands how heterogeneous and, they're rather evil beasts. But there are some really tried and true pathways that we know that these tumors can harness. And one of those has been the RAS pathways. KRAS in particular, we've known about it for over 4 decades, but it's been an elusive target for pharmaceutical targeting until recent time. There's a variety of reasons for that. Of course, we all stand on the shoulders of giants as we develop, these therapeutics, and I think we'll get into that in some of the questions. But Lumakras, it's an oral G12C inhibitor of the G12C mutation for KRAS. It's approved now in 45 countries around the globe. And really in these last 4 years, it's been a lightning fast program to get it to about, a little over 6,000 patients at this point. And I'll just end by saying that, this is a particular mutation that is found in lung cancer in particular. And these are patients who sadly, the therapies that are available today have failed them. And the initial approvals, of course, are as a monotherapy in those settings. And I think we'll get into more details, but this is really the beginning of the story. It's always fun to be able to talk about it rather than the end. But I think we can definitively now say that KRAS, at least the G12C variant, is a druggable target.
Thank you, Dr. Greg. Let's move on to Dr. Mark Confred, also Vice President, Hematology and Oncology from Insight Incorporated. Mark, can you please tell us a little bit about your product and the innovation that you're driving?
Yes, and thank you. I'm pleased to represent my colleagues at Insight in Wilmington, Delaware. I am going to be talking about our product pemigatinib, which is an inhibitor of a very important cancer pathway, cellular pathway called the FGFR, fibroblast growth factor receptor inhibitor pathway. Pemigatinib is marketed globally for the treatment of a rare form of biliary cancer as the drug Pemazyre. These are associated with alterations in the FGFR2 path. And more recently, it's received approval in the United States for a rare form of leukemia that's caused by cancer-causing rearrangements in FGFR1. Thank you, Mark.
Let's move on to Dr. Patrice Lee, Pharmacology and Toxicology Vice President at Pfizer. Patrice, please tell us a little bit about your product and innovation.
Thank you. Good afternoon, and thanks for you for having me. My name is Patrice Lee. I am VP of Pharm Tox and Exploratory Development at Pfizer Boulder, and I'm the non-clinical research lead for the Braftovi and Mektovi franchise at Pfizer. And on behalf of the Braftovi and Mektovi team, we are honored to join this group of panelists as one of the finalists for the Galien USA Award. At Pfizer, we work each day to help improve the lives of patients in need. With that mission in mind, in 2019, Pfizer acquired Array BioPharma, an innovative biopharma company in Boulder, Colorado, which has a impressive track record of successfully discovering and developing small molecules and targeted cancer therapies. I was part of the non-clinical team at Array BioPharma that invented Mektovi. With the intense competition in the pharmaceutical industry that existed back in the 2010-2011 timeframe, and few companies co-developing important therapies, Array established a partnership with Novartis, who invented Braftovi, to enable the development of the combination of these two drugs to address unmet needs for patients with cancer. So through the acquisition of Array, Pfizer was able to strengthen its oncology portfolio by adding the combination regimen of Braftovi plus Mektovi, which is approved globally for the treatment of BRAF V600E or BRAF V600K mutant unresectable or metastatic melanoma. As many of you know, BRAF mutations are very common in patients with melanoma, with about 50% of patients carrying such mutations. With the introduction of biomarker-driven targeted therapies, this has sharply changed the course of treatment for these patients living with unresectable or metastatic BRAF mutant melanoma. Braftovi and Mektovi are small molecule oral inhibitors of BRAF and MEK, respectively. So they target two different kinases in the MAP kinase pathway, which is also known as the RAS-RAF-MEK-ERK pathway. And mutations in the BRAF gene activate this pathway and stimulate cancer cell growth. So by targeting signaling at two different points along the pathway, Braftovi and Mektovi help to inhibit increased activation of the pathway that causes these melanoma cells to grow out of control. Since its initial approval by the FDA in 2018, Braftovi plus Mektovi have been prescribed to over 11,000 patients in the US. And today we are honored to see that this combination is being considered for this prestigious award. But most importantly, we continue to see the positive impact that these drugs have made on those living with this type of melanoma. Going forward, we look forward to investigating new indications for Braftovi and Mektovi based on this biomarker-driven approach to identify patients who have the BRAF gene mutations.
Thank you, Patrice. Dr. Kiran Patel, next, he's also Vice President, Clinical Development, Solid Tumors franchise at Janssen. Dr. Kiran, would you please tell us a little bit about the product and the innovation?
Yeah, sure. Yep, thank you, Sanjay, and thank you for the pre-Galien to nominate the Rybrevant amivantamab for the award for this year. Good afternoon, good morning, everyone. My name is Kiran Patel. I head up the solid tumor group in late stage oncology at Janssen. I've been at Janssen for at least 7 years now and have been really focused on coming up with new ways to target solid tumors. And the pathway that we have chosen in lung cancer has been very well known, the GFR pathway, and there have been a lot of innovation that has happened over the last decade. We still believe firmly that there's still a lot of areas to continue to improve upon, especially knowing that a lot of these patients with EGFR alteration do develop resistance that is driven through other pathways, such as the MAT kinase pathway. So Janssen took an approach of developing a first bispecific antibody that targets both the EGFR common mutation as well as the MET. And by targeting both, the intent is to not only just treat the disease, but also potentially avoid the emergence of resistance that is primarily driven through the MAT kinase pathway. And so Rybrevant, or amivantamab, it's called, is the first bispecific antibody approved in solid tumor, and is also the first targeted therapy to be approved for patients with non-small cell lung cancer harboring this very rare EGFR exon 20 insertion mutation, which is part of the EGFR family, but it has a different insertion that leads to mostly resistant to the available EGFR targeted therapy. So we're excited to share more at this meeting, and glad to be representing on behalf of Janssen.
Thank you, Kiran. Next and the last one I have is Dr. Susan Pandya, also VP Clinical Development at and Global for Oncology and Hematology at Servier Pharmaceuticals. Dr. Susan.
Hello, everyone. Thank you for having me. It's a pleasure to be with you all, and it's certainly an honor to have our drug, our baby, as I like to call it, Tibsovo, to be considered as one of the innovative products. So our story really dates back to the early 2000s when isocitrate dehydrogenase mutations were identified in a variety of different types of cancers, both solid and heme malignancies. And at that time, I was working at a smaller biotech company known as Agios that was more recently acquired in terms of the oncology portfolio by Servier Pharmaceuticals. And Servier Pharmaceuticals was founded in 2018, and it's a commercial stage, privately held pharmaceutical company launched in the United States here in Boston. So we're very lucky to now have the more sort of the nimble environment of a biotech like-minded company, but have also the support of Servier Group, which is the global pharmaceutical company that's governing by a nonprofit organization. The story with Tibsovo specifically is that we had identified IDH1 mutations in both hematologic and solid tumors, and our original proof of concept studies started in the 2014 timeframe. And this is where we were able to see profound impact in patients who had acute myelogenous leukemia harboring this type of mutation. And we were able to show that patients were able to go into complete remissions that were quite durable. And this was actually in the relapse refractory setting. So this was the first step towards innovation in heme malignancies. The next big step was in solid tumors, where there had been very little understanding about the role or the relevance, I should say, about IDH1 mutations. Similar to what Mark was speaking towards, biliary tract cancers are a rare, devastating type of disease that originates in the liver and outside of the liver. And over the past, I would say 5 to 10 years, we've started to really understand the genomic alterations associated with the biology of this aggressive cancer. And we've learned that IDH mutations occur in up to about 20% of patients with intrahepatic cholangiocarcinoma. So we were able to take our learnings from the hematology space and apply it to a high unmet need indication known as cholangiocarcinoma. And more recently, in the August 2021 timeframe, we were able to garner an approval, which serves as the first targeted therapy approval for this biomarker select population. So it certainly serves as a nice bridge between the hematology space into the solid tumor oncology space.
Thank you, Susan. Really appreciate that. So before we get into some questions that we have for the panelists, I want to tell the audience that there's a link that you could probably see on your screen that provides a bit more detail about each of the innovation that the panelists talked about, should you have interest to learn more. And that may also prompt you to ask some questions that you can directly ask the panel, that we have reserved time to present those questions back to the panelists. So please take time to go through the link and see if you have any questions about the product that was described, and then please let us know if you have any questions. So with that, what we want to do in the next phase of the webinar is ask questions to the panelists, and I have 3 questions that I'll ask each one of you. And in your introduction, you talked a bit about the product already. So this time when we have a little bit about the questions, if it can be a little bit brief so that we can cover the section that we want to cover in the webinar. So my first question to Dr. Greg is, why did Amgen choose to focus on the disease state and the indication that you have chosen? And in what way that represents the most, or the unmet need that was in your mind significant? And if you can tell us a little bit about that, that would be great.
Yeah, thanks for the question. We have some great examples of personalized medicine on the call here today. Of course, personalization isn't just about the drug, it's about the patient also, right? One of my favorite advocates says, I'm the person in personalized medicine, and we should always remember that. But, from the setting of Lumakras, of course, really the opportunity was created when some work that came out of Kevon Shokat's lab in San Francisco, which was just some wonderful structural biology work, showed that if you had this particular flavor of KRAS, the G12C, there was an opportunity to create a covalent inhibitor, take advantage of that C, and lock that enzyme or that molecule in the non-active conformation. And so that opened the door. And then, of course, I think some wizardry of medicinal chemistry then came thereafter to find a molecule that was drug-like and, again, could turn this switch off. But what was really at stake here was the fact that we knew that this G12C flavor of KRAS was present in 1 out of every 8 non-small cell lung cancer patients, or at least the adenocarcinomas. And it was known that this is very much, driving poor outcomes in these patients. So to be able to develop a molecule, if we could, that could turn off that pathway, the feeling was, though these are heterogeneous creatures, that was an incredibly high unmet need. There was no arrow in the quiver. There was no tool for physicians to then be able to layer into therapies. And of course, that's what came next.
Thank you, Dr. Greg. I think, Mark, same question to you in terms of why your company chose this disease state and indication and the unmet need that there is.
Yeah, so sure. So Insight is a science-driven company, as are the other representatives here, and the FGFR pathway is incredibly important from a biologic standpoint. It's intimately involved in cell proliferation, cell growth, cell differentiation. So it turns out that there are many tumors where you can identify cancer promoting alterations in these FGFR receptors, which are a family of receptors. There are several of them. So the insight story for developing pemigatinib is a classic example of medicinal chemistry. The molecule was discovered through rational design and iterative optimization of the lead molecule, and really thousands of molecules were synthesized and tested over a period of several years before we took it into the clinic. Pemigatinib is a receptor tyrosine kinase inhibitor. It's highly specific for these FGFR family of receptors, and it has good properties as an oral drug and can be taken once a day. So then we took it into the clinic in this form of biliary cancer, which as Susan has already highlighted, is an extremely bad prognosis disease. I mean, the patients we elected to study had all progressed after standard chemotherapy, and their survival really would be measured in terms of a few months. So this was a very needy population, and the results were quite excellent. The RECIST response rate, which is a rigorous regulatory standard for determining how well patients are doing with therapy, was close to 40%. But in actuality, over 80% of patients had some tumor shrinkage. So that's the standard that most practicing oncologists would use as to whether a drug is providing some benefit to their patients. And these responses were durable. And as a result, the drug is now approved in 39 countries for the treatment of these chemotherapy refractory patients with biliary cancer. More recently, we've taken it into the clinic for a very rare form of leukemia that's driven entirely by alterations in the FGFR1 receptor. And there the results are even more striking. And that's quickly led to approval in the United States, and hopefully we'll be able to make this drug available to patients worldwide. But there are many other kinds of cancer where FGFR is the driving factor, and we are pursuing these now in our development program.
Thank you, Mark. Patrice, same question, I think, about why you chose this disease state and the unmet need that you're seeing.
Thank you. Yeah, I mean, as you know, Braftovi and Mektovi weren't the first doublet to come forward for BRAF mutant melanoma, but when we looked at the unique profiles of Braftovi and Mektovi versus the other approved doublets, which have done wonders for these patients with BRAF mutant melanoma, we saw that encorafenib, at least non-clinically, has a longer on-target binding time, while Mektovi has a shorter half-life than the other approved drugs. So we thought maybe trying this different approach would add additional benefit for these patients who still at times are progressing on their disease. And at least from the non-clinical point of view, we were able to delay the emergence of resistance in some of these mutant melanoma models non-clinically, and we hope someday that will also, transfer to what we see in the clinic, because there still is a need, even though we are the third doublet that was approved, but we think we're a highly differentiated doublet.
Dr. Karan, if you could please also talk about a little bit about the.
Yeah, sure, Sanjay. Yes, I'll take a couple of steps back. As I think you're quite familiar, I think Janssen's vision is to cure cancer, and we are committed to pursuing transformational therapies to find cures and intercept disease in patients with high unmet medical need. And so from that philosophy, especially in solid tumors, targeted therapy still remains a cornerstone of trying to find ways to treat and intercept and treat the disease. So that really has been the focus for Janssen for several years now. Amivantamab or Rybrevant is a great example of that targeted approach, using a different platform perhaps, rather than small molecules. There are ways we can also target using large molecules to be able to precisely help this group of patients. And then lung cancer, of course, is a very significant high unmet medical need. I think the group here and the audience knows very well how devastating the disease is. And going after a population that we know has not benefited really drives the development programs where we could potentially show where others have not benefited. And if the drug like Rybrevant can provide that benefit, it provides us additional confidence that, there are ways we could potentially have this drug also being expanded and developed, not just only in EGFR mutant, the common EGFR mutant non-small cell lung cancer, but also in other tumor types, where there could potentially be an EGFR overexpression and in EGFR wild type population, as well as in other tumor types. So we're quite excited about this, but really it kind of falls into the core philosophy of Janssen of really going deep into understanding the targeted therapies and then identifying ways to target those specific population.
Thank you, Karen. And the last one, Susan, if you could also answer the same question, which is, why this indication and disease state, as well as the unmet need that you see here?
Yeah, absolutely. So Tibsovo perfectly fits into the portfolio and the mission specifically that Servier espouses to, which is really to tackle hard to treat cancers, even if they are rare. And I think that's really important because as we start to think about these biomarker select populations, the population gets smaller and smaller. But that doesn't take away the devastation associated with the disease. So specifically in cholangiocarcinoma, as I mentioned, it's a rare type of disease, and especially because standard of care genomic profiling wasn't really the mainstay of how we identified these patients earlier when we were developing Tibsovo. It was really important to have a strategy for our development that would meet the needs of patients and be able to pre-identify these patients in a pretty quick manner. So I think this is an area that deserves a lot of attention when we think about precision medicine, and it's something that Servier is very focused on, helping to harness the effectiveness of our compounds and expand it across indications. Tibsovo is a small molecule inhibitor of the mutant IDH1 enzyme, which is involved in cellular proliferation as well as differentiation, and can impact a variety of other cell signaling pathways. So our focus really has been to not only show the efficacy in the heme space, but also solid tumor space. And we were able to quickly move from phase 1 proof of concept study into a global randomized phase 3 trial, which represented the first randomized study in this disease indication for a biomarker select population. The trial results basically showed a 63% reduction in the risk of progression or death, which was a huge step forward. And this was already in a pre-treated population, so in patients that already had a pretty poor prognostic outcome, as Mark was alluding to, in terms of survival measured on the order of months. So to be able to have that kind of impact in a pre-treated setting really shows the impact of this therapy for these patients.
Thank you, Susan. And I think after listening to all 5 of you, it's very clear why these products were nominated. It's evident in terms of the disease area and the unmet need, as well as the clinical outcomes. So I think this is great. Let's pivot to talking about the celebrations that you've had with the science and with the outcomes that is there. Let's get to the challenges that your companies confronted while developing this, going from early proof of concept through the commercialization and then now post-commercial. So I'll start again with Greg, with you, to talk about just the converse, that, great product, great innovation, helping patients, but then what were some of the challenges that you saw developing this and commercializing it?
Yeah, drug development's hard, right? This isn't math, this is biology. And so, there's a whole long list, but I'll hit some of the challenges that I think are probably, shared in many cases by all of us. Once we got through the chemistry angles of this target and, the drugging the undruggable, approach, I think we knew that we had an active molecule on our hands. We knew that the safety profile was one that seemed, certainly different than chemotherapies, but one that was going to be tolerable for this patient population. And then the question was how to get it to patients as quickly as we could. As I mentioned, the first patient was dosed just about 4 years ago. So this was a sprint. Again, when you sprint at that speed, of course, you have to make decisions along the way based on not gigantic sums of data, but you have to be smart, work with physicians, work with patients, certainly work very hand in hand with regulators. And I think the challenge of doing that and maintaining the highest quality data sets, of course, and by the way, we had a world pandemic during the time as well. Again, these were, overcoming come through tremendous work of people, not only at Amgen, but of course, the patients who enrolled in the study, the physicians that we worked with around the globe. This was a program run on 5 continents, right? And so from that standpoint, being able to pull that together, move forward, and again, let's not forget the willingness of the regulators, not only in the US, but around the world, to work very closely with the sponsors to make sure that, we were keeping our eye on what was important. We knew that the patient need was there, but we weren't compromising any of the quality of the data sets along the way. I would say that was one of the, major challenges, working at that speed. We used to have 8-10 years to ready organizations, ready the world for these therapies. And in this fast-moving era where patients, again, need these therapies, that was something that, I'm quite proud to see that this group of, multiple stakeholders was able to come together and make that happen.
I think pandemic clearly was challenging. So congratulations that you were able to overcome some of those challenges. And Mark, same in terms of was there anything unique or different from what Mark said that you experienced in your organization?
So we were fortunate to have conducted our registration study before the COVID pandemic, which avoided many of the complications that we now see in our present day studies. But I think what I'd like to emphasize is that what I do as a clinical lead is building on the shoulders of the early development and the technical people who have made it possible to identify a candidate molecule through as I mentioned, screening thousands of candidates through the chemistry processes and turning it into a pill that can actually be given to people in a safe manner. This is years of work, and once this has all been accomplished, it's a relatively straightforward question to answer in the clinic in patients who really have no other good options, whether the drug works or not. And we were fortunate to have the support of the regulatory agencies. I mean, we had FDA gave us orphan status because this is a rare disease, and breakthrough status, and priority review and accelerated approval, all of which just emphasizes how important it is to be developing drugs for these rare populations with no good therapeutic options. And I think the regulatory processes actually work quite well in this setting, and we hope they will continue. But when you have a drug that obviously is helping patients, then it's a straightforward matter to get it approved. And this is not just a U.S. focused path either. I mean, we view the patients of the world as as needing therapy too, and they're our customers as well.
Thank you, Mark. Patrice, anything specific to Pfizer and what you faced?
I think I mentioned it when the answer to the prior question. As you know, I mean, this is not the first doublet that was approved. So the onus was actually on me and my team to generate, a more comprehensive, non-clinical data package to actually explain the differences of Braftovi and Mektovi versus the prior approved doublets. I mean, this was a welcome challenge. We generated the data, but I just think we had a little bit more work to do to convince people that these two drugs are really different than what was already approved.
Got it. Thank you. Kiran?
Thank you, Sanjay. I think, this audience is very familiar with drug development. It's probably one of the most complex things, out there. And even if you've done it several times, every time is a new experience. And developing amivantamab was no different either. This was the first bispecific antibody that was discovered. A lot of work was done at Janssen. But knowing that, there could be a difference in terms of the affinity to one to the other because of the underlying on-target toxicities, such as EGFR-driven toxicities, like rash and diarrhea, and similarly for the MAD-driven toxicities such as edema. It was very critical to, identify the right affinity for both arms so that we can balance out, the safety, but then also be able to see the activity. I think that probably was one of the highlight or the biggest challenge, from the discovery front, that we were able to overcome, quite early on. And then I really concur, with Greg. I think COVID really kind of changed the dynamics and maybe even made it, the complex things we do even more complex. And just a simple thing like a challenge with getting vials, it really affected all the drug supply that we were providing across all of our centers across the globe, and that led to some other challenges around identifying different ways to provide the drug. And so those are examples. But I do want to highlight one thing, is that actually two things. One is that I do feel that COVID-19 pandemic really helped us to see how we can work together, academics, industry, pharma, biotech, regulators. It really came together to really join the forces of how we can work together and work in a way that ultimately help our patients. And I think that was a great testimony of just knowing the development of COVID-19 vaccine, but also the work that went behind, taking care of cancer patients, was just amazing to see how everyone came together, flexible, allowed patients to stay on therapies, identify ways to continue to treat them despite, a lot of hospitals were shut down. Giving IV drugs is not that easy, when they have to come to the clinic. But despite all those challenges, we came through really with a lot of lessons learned, and I hope that we can continue to advance and try to bring, these medicines even faster for our patients. COVID-19 can be approved in, 15 to, 18 months. I think we can do the same for a lot of other drugs that really are desperately needed for our patients.
That's right. Karen, thank you. Susan, any last comments on the challenges that are different from what has been
others have
before?
commented about the complexity of the chemistry and making sure that it's optimized, and in our context also being able to have a measurable biomarker for suppressing a mutant enzyme. This, as a leader in the metabolic oncology space, this was the first in class product that would be targeting a mutated enzyme in cancer cells. So a lot of the work in the preclinical space really laid the foundation for how we would then be able to select the appropriate population within these different types of disease entities, so within heme as well as in solid tumors. So I would say one of the challenges was that we were going into indications where genomic profiling did not necessarily have an IDH mutation, test on the panel. So it's very important in these rare disease settings to be able to early on start developing clinical trial assays with vendor support to be able to select the appropriate population for your first early proof of concept study so that you can then move quickly into the randomized controlled setting. And this is also important because companion diagnostics are usually part of what needs to be coupled with the drug that's being evaluated in that specific indication. So that was a challenge to some extent, but it was one that we prepared for. I think in terms of recruitment, we too were able to recruit our trial globally before the pandemic in a reasonable period of time, about 2 years. And one of the efficiencies that we thought was really important to incorporate in our strategy was to allow for pre-screening consent. So for patients receiving first-line chemotherapy, for instance, they could have their tissue tested just to see if they had the IDH mutation. And if they did, they would then have the opportunity to go on to our trial if and when chemotherapy stopped working. I think that was also a smart move because it allowed for patients to then think about, okay, what is the next step for their therapeutic options? It also helped us with recruitment significantly. There's so many other challenges I can speak to, but I think that one of the main ones, and others have alluded to this, is really partnering with regulatory agencies earlier, which we successfully did, as well as patient advocacy groups. Because again, when little is known about the disease and little has been published, it's really important to sit down with patient advocacy organizations and design a study with them in mind and with their voices represented. And so we took those steps very methodically and deliberately. And I think it really allowed for us to have a very successful outcome.
When did you engage with patient advocacy groups? So the early?
Yeah, so we did. Right after our phase 1 study results were becoming more mature and we were ready to make a go into the late stage program for a randomized controlled study, we actually presented our trial design to a US-based advocacy organization and got direct feedback. And we altered the study design based on that feedback, which in the end I think was the right move. There are always trade-offs when you're making these types of decisions, but I think it was the right move. And again, the study was designed with patients in mind. And FDA was very aware of that when we presented our final trial design to them.
That's excellent. Thank you, Susan. I want to point out to the participants on the webinar, I think I don't see any questions just yet. I want to remind you that you have an opportunity to ask the question directly to the panelists. So please take a moment to write some questions for the panelists. And in the meantime, as we are waiting on those, I'm going to open this forum up a bit. Instead of going individually to each one of you, I want to have a group discussion. And it's really about the innovation in your space. As you see, what's coming next in your space, in your disease area, or the indication that you're working on. And any one of you can start the conversation, and then we can go from there. I have some subsequent questions as we go from that first initial set.
Yeah, I think the adoption of a tumor agnostic approach for future clinical trials is going to, blow the space wide open, so to speak. The FDA just issued guidance for industry about a week ago on how to do these trials. So the challenge will be we'll be able to have the companion diagnostics in place. We'll need RNA-based testing and various other things, but I think that's going to open up a lot of opportunities, especially for these highly targeted therapies to go into any tumor type and early in clinical development.
Just to add to Patrice's comment, absolutely agree. I do think that there's a complexity in addition, though, that of course continues to be a really hard challenge in solid tumor. Whether it's BRAF or we're talking about RAS, there's contextual importance of the lesion in different tumors as well. And so, part of me thinks that, as we get better tools, we'll be able to target more personalized therapies. I think targeted protein degradation is a fantastic approach to open up new target space. But on the flip side, thinking about the patient as well, I hope that, 10, 20 years from now, we're going to have better tools for general pharmacodynamics, whether it's, in heme tumors, we have minimal residual disease, something similar in solid tumors, or, whether I think imaging probes have unfortunately been too much of a blunt instrument, but some ways to, again, understand the biology of the patient, not only pre-treatment, but in real time, that doesn't require, an 18 gauge needle going into a selected, portion of the tumor. I think that, once we have, that ability to actually measure what's going on in a patient in real time, it'll make our studies very difficult to do. But on the back end, I think that, there's no way around this complexity. We get fortunate and find these Achilles heels every once in a while, but in all likelihood, I think, tumors got the way they are by being these, adapting organisms. I think we're going to have to adapt our therapies in an individual patient. I don't have the answer to that, but I do think, again, having more tools is great. But at the end of the day, we'll also have to think about the way that we approach the individual patient. And it's going to be hard.
Yeah, I would just maybe concur with that, but also maybe just kind of two different aspects, right, of innovation that I think is already happening, but it probably would look quite different 10 years from now. I think one is the approach of going for different multiple platforms, and we have good success in heme malignancies. We're not there yet for solid tumors. The technologies that actually have transformed, care. And I do think that there are complexities with solid tumors. We have a huge tumor microenvironment that, makes it really challenging to give drugs. But I really hope that that's where, a lot of innovation would happen, where we could potentially come up with different platforms and technology that allows us to go for a combination approach. And I would say that number two would be really going into early disease. Disease is complex, and I think it gets more and more complex with heterogeneity, with various alterations. And so trying to go into early disease and intercept the disease early in their evolution potentially could avoid having this multiple pathways that are getting activated. So and there are complexities there. Of course, you can have a drug that has a good safety profile that you can go into early disease, but we also have to come up with unique endpoints that would be acceptable to regulators. Studies that going into early disease could take 15 years to mature for the endpoint. So there's still a lot of work to be done, but I would say those are the two areas that perhaps would be really key for us to transform the solid tumor space. We can come up with drugs that can really help our patients.
I would just add, I think we're in an era where you're seeing a lot more pharmaceutical development and, companies collaborating effectively. You're also seeing both large and small companies harnessing each other's individual capabilities and be able to work effectively. You see these types of collaborations all the time. So I think that is also a major advancement for innovation. I do think that, if we can start to work more towards predicting the genetic landscape and the genetic evolution of these different difficult to treat malignancies, whereby you may have a driver mutation, but then there's an elaboration of a variety of other genetic alterations. So trying to get one step ahead of where the cancer is, I think, is something that we should spend a lot of time on. And I know several groups are already working towards this. So, I think we're in a place where we can do a lot for patients, and there will likely be more of a tumor agnostic approach, as others have alluded to, as we start to unravel the complex biology of these different disease types. So I think we're in a golden era, frankly. This is a really exciting space to be in, a lot of complex, challenging questions and challenges at large, but at the same time, we now have the tools and the technology that we didn't have before.
Yes, I agree. It is a golden era to be living through and participating in. We all love our jobs for that reason. But there are some trends, and I think regulators and to a certain extent the public are asking more of us in the development process. And specifically some of the questions which might typically be answered once the drug has been approved are now expected to be answered in the development process. And while these are not unreasonable things to ask, it does require more work of us, and will result in longer times to bringing new therapies to needy patients.
Yeah, no, that's excellent. I think if I were to stretch this and paint a future scenario, like 5 years from now or even 7 years from now, what kind of innovation do you think this same forum, Galien Awards, might be reviewing? You think that if we were to fast forward, like 5 to 10 years from now, you think fundamentally different innovation that might come than where we are today, given the advances that you're talking about, the tools, technology that are available? And if we were to eliminate some of the impediments that we may have on the data side, accessibility of the data and analysis of the data infrastructure, then what would you predict if you had a crystal ball from now, like 5 to 10 years from now, if you were to come back to Galien, and what might be the headline?
Just a quick thought. I mean, I do think 5 years isn't very long in drug development. So probably things need to at least be within eyesight if we think they're going to be in 5 years. We mentioned targeted protein degradation again. A lot of target space that could potentially open, but it's got to work. On the flip side, I think we are seeing the bispecific antibodies are doing more exciting things with their biology now. We have the CD3 bispecifics that are trying to be an off-the-shelf T-cell redirector. I think in solid tumors, we're going to start seeing more and more. In heme, we've seen great progress, particularly on the lymphoid malignancies. But I do think that these multi-specific drugs are going to be what we're going to see more of. They're going to get more and more clever, I hope. And I hope that'll open up not only more warheads against the tumor, but also more selective targeting. I think it was Patrice who mentioned on the antibody, I'm sorry, it was Karan who mentioned the targeting of affinities and fine tuning of antibody affinities. Actually, Patrice, it may have been you as well. But for small molecules, but nonetheless, that sort of work, I think we're going to see more clever uses of technologies we have right now by stacking them on top of each other and thinking about using them in different sequences.
Yeah, I mean, I do agree. And I think the targeted approach is not going to change, right? We have to keep targeting, the tumor, right? Where, our focus is. But I think how we target has really shifted and, going from oral targeted therapies to, going for targeted, approach with ADC platform really speaks to, we continue to, progress, with, oncolytic virus, for example, again, trying to get to, the tumor where we really can have the biggest impact. But I also do think that, there will be an approach where you go for that, you go for targeted approach, you combine that with other immuno-oncology programs that allows you to, have your body harness, the immune system that perhaps, would last longer than just, treating something locally. And then some of this, radioligand approach, I think is really, coming and really, demonstrating, some really good, benefits. Again, the approach is, how do you kill the cancer cells that it doesn't have the ability to continue to continue to grow, and drive resistance. And so I think all these approaches are going to really start panning out. I also feel strongly that, at some point there will be a combination of drug device approach, right? Where, especially when you go into early disease, you got to really find ways to target, by, instruments that can allow you to get to a specific area. So a drug device approach could be something I think, could be quite interesting, Sanjay, for this award, because, that will be a unique way to, treat and potentially cure cancer.
My hope would be, and I hate to say a crystal ball because I don't want to jinx my own programs, but I think one of my hopes would be that we've learned from the pandemic and that we're able to operationalize studies in such a way that we can get faster readouts, faster recruitment, right? So while I absolutely agree 5 years is short in the development lifespan of a clinical compound, my hope would be that operationally we're able to fine tune along the way so that we can tap into patient populations that have not traditionally been represented in clinical research. Because that's a well-known challenge. Much of what we've talked about, the rarity of the disease is one piece, but also tapping into the patient populations that can't drive into major disease, tertiary medical centers, that can't take time off of work. So how can we have a decentralized process whereby we're able to engage patients out in the community who can participate in clinical trials and still collect meaningful data and have an impact. So I think that that's one area that I'm hopeful in the next 5 years we'll be able to move the mark on. With respect to innovation and drug development specifically, I would hope that we make a bigger play with the diseases and the biologies that we're already talking about, and that we don't rest on our laurels, that we understand that there's more to learn. And I think that's where having ongoing translational efforts to think about resistance pathways, or in the case of our program, moving ahead in brain tumor research, these are areas that continue to require a lot of thought and planning. And I'm hopeful that we'll be able to make major strides in the coming years.
Yeah, and just one last comment. One thing that we're doing, at least at the Boulder site within Pfizer, is we are focusing on brain penetrant drugs, simply because that has been a space that people have avoided for so long. And it's not just primary brain tumors, it's metastatic disease from other melanoma or lung and what have you. So we are focusing on that. And that's a big challenge because it's a different safety paradigm too. So I see that coming in 5 to 10 years. And we mentioned tumor agnostic approach. I think more people are going to be doing that, but you're going to have a different tumor microenvironment with each tissue now that you probably haven't really looked at very hard to see if there's something else that you need to target. And so that's going to be an exciting time.
Yeah. And I think the other thing we some of you have mentioned about, I would talk about the enablers of science, right? So regulators, for instance, have become a lot more, they're no longer, FDA is no longer draconian or anything. They've been very facilitating innovation and science. So I think we're seeing more and more how regulators are working. And Susan, you talked about the collaboration between the pharma as well as with the academic medical centers and others, the tech transfer that's happening. Would you also collectively think more pharma collaborations in a pre-competitive way that may also foster science and faster and accelerate some of the innovation that we see?
Yeah, and I think that many of us on this panel have seen several examples of that, large biotech, or excuse me, small biotech with large pharma or two large pharma companies working together, especially in these hard to treat malignancies and in settings where you can't do 5 different trials, or you can't necessarily go from phase 1, phase 2, 3 in the sequential way. And it's important to really collaborate quickly and move quickly into these disease spaces. So we've seen it, certainly with Servier, we have several collaborations that are ongoing. So I think that it's more primed, I think, the ecosystem that we're working in right now than it was traditionally. But I'm sure others can also comment, having worked in different disease areas and within different global organizations.
Yeah, yeah. And we certainly see the physician and the hospital system really being very proactive and collaborative in the last few years. So that's certainly happening. One last thing, and I think, moving a little bit away from the clinical development, more on the manufacturing and CMC, to be able to take these therapies, these at scale. So one thing to be able to do the science and to be able to have the druggable candidate, but can we actually manufacture at scale to treat population at large and not just the rare diseases? Any comments that you may have around what the future might bring from manufacturing challenges that you may be aware of, and what your companies might be doing, or things that we need to do more?
Yeah, I think that's a great question, and it's pretty complex, right? Because drug manufacturing has really transformed right over the last decade. With something like a small molecule to going for antibody to now bispecifics and CAR-Ts, and so. I think it's really challenging. I would say probably for the entire industry to continue to move in that direction. But I do think that there is a clear reason for more investments into that, because I think a lot of companies, Janssen included, and others, small and big, probably have the same challenges that we have. And coming out of COVID-19, I think it probably got amplified even more. But I would say probably, the infrastructure is critically required. I think the expertise is very unique to a lot of the work that is being done. So it's not something that can be just, parsed out and expect to, be, precise. In some cases, even the delivery of this drug has to be done within a certain period of time. So it's really very precise manufacturing process. And it probably requires a lot of infrastructure, investments, to continue to keep up with the evolving science.
Yeah, and I think we clearly, I think in a few years we will be able to overcome some of those challenges. What I think, because as these gene therapies are now going on with the solid tumors, we're going to be treating, or need to treat, a lot more patients than we have been treating so far with the heme-based CAR-Ts. So we need to address that manufacturing challenges, and I see in the next 5 to 10 years we'll be able to get over it.
Yeah, at Pfizer, at least we involve pharmaceutical sciences and CMC teams really early, so they can be partners with us even before we file an IND, because they need to look at routes and decide if there's big hurdles and identify CROs that can do starting materials. So earlier the better, because you don't want it, the onus hanging on them in the end when they say, "Oops, we can't do this."
Yeah, Patrice, just to highlight again what you're saying, that in many ways, I think some people call them tech ops or pharmaceutical sciences. These are the unsung heroes that work often behind the scenes. We get to give the presentation at Galien, but they slave away to give us, take 1,000 molecules and pick the one that we ultimately bring into the clinic. And so, if any of you are listening on the line, thank you for all you do. You're an incredibly important part of this process.
Very good. I know we are at time almost, and I don't see any questions from the participants. And with that, I want to thank you all 5 of you individually. Thank you for taking the time. I know you're all extremely busy, but had the opportunity to listen to you and the participants on this call to talk about the innovation that you're driving and the product that's impacting the patients' lives. So thank you very much for the work you're doing, and thank you for your participation on this webinar. I appreciate it.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye.